Welcome everybody to our October 13th meeting of the IMAG-MSM Working Group on Multi-Scale Modeling in Viral Pandemics. We appreciate people being here. As always, I need to remind you that the meeting is live streamed and recorded, so please be aware of that. We have a single presentation by a pair of speakers today, which we'll come back to. Tom and myself are always available for your suggestions or comments. Jim, Lorenzo, and Joel are also available to help out. I believe Lorenzo is traveling today, so he's not here. I know Jim is traveling today. As usual, we have our communications channels. They only work if you use them. Please do help us distribute the announcements. We have our archive of YouTube recordings. There are more than 130 talks now on that archive. We could use help distributing that material. It's really a great resource in the community. We have a Twitter feed, MSM Viral. Twitter only works, I guess, if you're Elon Musk, but please help us with that. We're going to have a second-year anniversary. It's officially next week, slightly off by a day or two next week. We were going to do a discussion of the future next week, but for a variety of travel and other reasons, we're moving that to the 8th of December. We could use some help updating the IMAG-MSM wiki page to reflect the content. It shows a lot of the papers and grants that have come out of this working group, but we would love to have more help on that. The slide deck is available for editing. If you would like to edit it, please let us know and we will provide access. If you want to just tell us what your relationship with the working group is, collaborations, funding, publications that grew out of talks or other activities of the working group, we would very much appreciate those so we can put them together for that December 8th meeting. Are there any announcements from the room that people would like to make before we go forward? I think, Reed, you and Grace had something that you wanted to put forward that I think is on the next slide, which we'll come back to. As I said, the anniversary meeting is postponed to December 8th. The steering committee meeting is going to be an optional tomorrow. We're going to be working on the slide deck for the anniversary meeting. If you'd like to come help us, you're welcome, but that's not required. Any other announcements before we go on? There's a letter from Grace and Reed, and maybe I should let Reed introduce that for us.
Sure. Let me look at the slide here. So yeah, so long story short, the IMAG, as you all are aware, is this interagency modeling and analysis group, and this is one of the subgroups of the steering committee, which was formed long before my time. There's a number of folks across the community, and they made a mission and vision statement, I think in a, like at an in-person meeting on the back of a napkin at one point. And I'm being slightly tongue-in-cheek there, but they put some effort into revising it, and it's not perfect, but we needed to get something out. So we sort of posted it on the wiki, and there are ways to comment on it. So you can take a look at it. A number of folks on this call have already commented on it. You can go to that link. You can take a look at it. You can say what you like about it, what you don't like about it, grammatically or whatnot. You can reply. There's an email you can reply to, an email box that I monitor with Grace, or you can post your comments directly on the wiki if you are part of IMAG. And if you are not and you want to become so, you can send a note to that box, and we'll get you set up. So I think that's that. And then I think the other quick announcement is that there is talks in the works for a meeting at the NIH main campus, June 28 and 29 of 2023. I hope I got those dates right. And I think the details of which are still being worked out, but it's sort of a broader IMAG meeting. This might have been mentioned on here before. So though obviously this group would be the first to know of updates to that and the details of that, but they're still being worked out. So those are my two updates, I think. Yeah.
Okay, thank you, Reed. We have a pretty full schedule coming forward. I know it's been a little sparse the past few months. We have, again, next week's talk and discussion is rescheduled. October 27th, Julio Saez Rodriguez from Heidelberg, Russell Schwartz from Carnegie Mellon on November 3rd, two speakers from Physique on November 10th, Claudio Montero coming back from Nova Discovery. We had a great talk from Nova Discovery some time ago, updating us on their software platforms this time, their clinical trials, virtual clinical trial software platform framework. On the 17th, no Thanksgiving talk because we're sufficiently US based that we won't do Thanksgiving. Two talks at the beginning of December. First, the rescheduled anniversary discussion. And then on December 15th, we have Maria Abu-Chakra from the University of Toronto, who's going to be talking to us about mainly Canadian initiative for medical digital twins. And I was at the kickoff meeting for that in Vancouver last week. And that's growing out of the Canadian Stem Cell Association, trying to build detailed mechanistic multicellular models of induced pluripotent stem cells in organoid culture. I think that's a very interesting initiative, and look forward to hearing from Maria about that. As always, please suggest speakers. We have plenty of people here to try to track down speakers. We have our tiny URL available so you don't have to type so much, and we'd appreciate your help getting those people on the schedule. So with that, people know I don't do elaborate introductions. We have two speakers today talking about MRSA and what we can do to predict it, and maybe a little bit about what we can do to deal with it. And so I will turn over the meeting to you.
Thank you so much. We really appreciate the invitation and pleasure to join you today. Are you able to see the slide deck now?
Yeah.
Okay. So today we wanted to kind of provide a succinct overview of the work we've been doing on this area. And it has to do with systems immunobiology and tensor modeling of persistent MRSA bacteremia. My name is Michael Yeaman. I'm professor of medicine at UCLA and chief of the Division of Molecular Medicine at Harbor-UCLA Medical Center. And Aaron, I'm sure you can introduce yourself now or when you begin your slides, but why don't we just move on? I do want to say that we really appreciate the support from the NIH for the U01 upon which most of these findings are based. So briefly, what we hope to do today is touch on about 5 points. The first is the problem of persistent MRSA bacteremia. Another is a quick review of the complexities of host MRSA interactions, a few highlights of the systems biology U01 and some of the key findings that have emerged. Then really the feature part of this discussion will be Aaron's summary of the tensor modeling of MRSA bacteremia. And then we'll come back to a couple of summary points and prospectus. So with no further intro here, let's just talk briefly about the problem of persistent MRSA bacteremia. In a nutshell, MRSA is a leading cause of bacteremia, as well as other endovascular infections, including infective endocarditis and device-based infections. Up to 30% of MRSA that access the bloodstream fail to clear using gold standard therapeutics. Those include vancomycin and daptomycin. These isolates are considered susceptible per CLSI antibiotic breakpoints in vitro. So there's immediately this dilemma between seeing in vitro susceptibility, but they are not susceptible in vivo, at least in context of the blood. These particular isolates are called persistent MRSA, and they are associated and may even drive worsened outcomes, including death. And I'll just point out that persistence differs from classical resistance, which is usually strain-specific and structural gene related. So there's a structural gene encoded mechanism of action, mechanism of resistance. It's also different from tolerance, which is a population or heterotypic type of lack of susceptibility that really could be summarized by the word dormancy. In comparison, persistence is an active response, but it tends to be transient and only in vivo. We'll maybe touch on that a little bit. So again, briefly, some of the complexities with respect to host MRSA interactions are summarized here as regards endovascular infection and bacteremia. So when staph enters the bloodstream, there is, of course, usually some sort of experiential immunity. So most humans have encountered staph many times in their life and may even have the organism colonizing their skin, their nose, or other areas of the body. So there is a pre-existing adaptive memory. However, many humans are susceptible to recurrent staph infections. And so there's a little bit of an uncertainty between immune response and protective immune response. And we'll touch on that as we go a little bit as well. When the organism enters the bloodstream, there are, of course, immune effectors present, neutrophils, platelets we know have direct antimicrobial functions, as well as antibody. And yet the organism tends to find its way to bind to endothelium, begin to evade early cellular features of the immune response, and with proliferation and invasion, then establish infection and abscess that can lead to deeper infection of parenchymal tissues. Ultimately, because of quorum sensing features that the organism can execute, at a time when the abscess achieves a high enough density of organisms, these organisms will undergo quorum sensing. They will turn off their local virulence factors in favor of virulence factors that allow them to escape, move back into the bloodstream, and kind of repeat this process over when they find another site. So that's a little bit of a working model of the life cycle of persistent MRSA bacteremia. From a systems perspective, we like to think of persistent MRSA bacteremia as an intersection of three systems. The first is the organism and its ability to adaptively and dynamically respond to changes in environment or threat. Of course, the host immune system is another system in itself, and antibiotics superimposed upon these two systems create sort of a third dimension of this interaction. So our UO1 really focused on this intersection between the organism, the host, and antibiotic therapy. The key hypotheses that we took forward into the UO1 are briefly summarized here. The first is that persistent MRSA bacteremia reflects a uniquely in vivo phenomenon that cannot be fully recapitulated in vitro, at least not to our knowledge as yet. In some ways, we could summarize this phenomenon as an adaptive response of the organism to antibiotics in context of the in vivo condition. Also, adaptive response of the organism to the host immune system. And then the third dimension that's not always as well appreciated is the response of the immune system to the antibiotics. So we have this sort of three-way intersection that we feel is important to understand better and was the focus of our UO1. Another hypothesis was that host pathogen signatures of persistence can be defined and modeled, and that's what we'll talk a little bit about today, and that systems approaches can enable predictive accuracy that can look into the future of infection and course of infection, and thereby perhaps benefit clinical intervention and improve efficacy and outcomes. So just to summarize the background of the U01 structure, and in particular, the importance of the resources that were used, I just want to emphasize the Savage biorepository at Duke University, led by Vance Fowler, which was integral to this work. It was essentially enabling of our assessment of over 3,000 cases that Vance has accumulated there. And then we sort of established inclusion criteria and exclusion criteria so that we could really try to identify the most useful cohort of patients to study with respect to identifying host pathogen signatures of persistence. So briefly, the inclusion criteria were individuals who had MRSA bacteremia and were culture positive, who had an isolate available, particularly for persisting cases who had multiple longitudinal isolates, that there were clinical metadata available, that the treatment regimen was vancomycin, and these were adult patients. We excluded individuals who were culture negative, who did not have an isolate available, did not have completeness in clinical data, were being treated by non-vancomycin regimens or multiple antibiotic regimens, who were neutropenic or had other immune suppressing features, who were outpatients, had polymicrobial infections, or were pediatric cases. So this algorithm here just shows we had 3,000 cases to work with. As we went through these gatings, we had identified 300 cases which were really ideally suited for matching. And with the help of David Jurtson at UCLA, who's really an excellent biostatistician and epidemiologist in many respects, we identified a number of persisting bacteremia and resolving bacteremia cases that were as well matched as we could hope with respect to demography, time points, and controls. So we took these cases, and these were individuals from whom DNA and RNA, serum, PBMCs, and the isolate were available. And then we studied these with respect to immunogenomics, including transcriptomics, proteomics, and then modeled the data through various approaches, including what Aaron will talk about in a few minutes. In parallel, we did introduce the persisting and resolving organisms into various models, including mouse models and rabbit models. We won't talk about that today, but it is a very interesting parallel to the human clinical studies. I did want to mention briefly that the U01, in addition to the work that you'll see described in just a moment, really was designed and succeeded as a launching pad for a number of what we consider to be rising stars. The work that I'm going to summarize in just a moment was really largely led by these individuals who are all kind of at early stage investigators, and we're very fortunate to have had them working with us. So I just wanted to highlight 3 studies that came from the initial part of the U01 that kind of reflect the impetus for what Aaron's work has led, and we'll come to that. The first was this paper, which focused on protective immunity in recurrent Staph aureus infection. And it really emphasized 2 themes. One, that there is localized immune signature specificity, and that there is innate memory conferred by macrophages. So just to summarize that, again, as I mentioned, susceptibility in humans to recurrent MRSA infection occurs despite acquired or adaptive immunity. So we all have antibody to staph antigens. We all therefore have B cell and T cell knowledge of Staph aureus, and yet many people get recurring infections. And there's something about that that should teach us that adaptive immunity may not be the only protective mechanism. We also found that immune responses to MRSA differ in distinct temporospatial contexts. in many respects, you could summarize this as if you put Staph aureus in any number of different tissues of the body, it is recognized and responded by the immune system as though it were a different organism in each place. So it's a very heterogenic immune response. And importantly, we also found that there is strain-specific, likely antigen-specific, innate memory encoded into macrophages that appears to be crucial to protective immunity. This differs from trained immunity, which is not specific. So we believe this goes one step further and establishes antigen-specific, or at least strain-specific, innate memory. And whenever we talk about innate memory, we start to think about epigenetic encoding. And that kind of was where we went to the next summary point here, and that is that there is genetic variation within humans that can discern persisting versus resolving likelihood in terms of MRSA bacteremia. So this paper, Genetic Variation of DNA Methyltransferase 3 Contributes to Protection Against Persistent MRSA Bacteremia. We found that human persistent bacteremia versus resolving bacteremia cases, even though ideally matched for demographics and comorbidity, revealed a SNP in the DNA methyltransferase 3A gene. This was an adenine to cytosine genotype change. And this particular SNP promoted CpG hypermethylation. And in particular, this genotype, by hypermethylating transcription factor sites and enhancer sites proximate to IL-10, was associated with a reduction in IL-10 levels, and that in turn correlated with greater chance for resolving bacteremia. And then lastly, again, just another highlight is that in direct examination of the DNA methylation signatures in humans with persisting and resolving bacteremia, we found that there were differences sufficient to discern these outcomes. So methylation patterns of host DNA were able to discern persistent versus resolving outcomes with a pretty high degree of confidence or accuracy. The differentially methylated sites were intensified relative to transcription factor or enhancer sites. And interestingly, there was hypomethylation of STAT1 pathways in persistence as compared to resolution. And just to give a little bit more color on that, STAT1 is often a pathway that controls interferon gamma-like responses. And so if there's hypomethylation of STAT1 pathways, you can imagine a de-repression of interferon gamma. And because interferon gamma can counter-regulate IL-17, we have this potential suppression of IL-17-mediated pathways that are in many respects believed to be important in controlling staph. So again, there's this multiple dynamic balance among T cell and other pathways that are likely important in determining outcomes of persistence versus resolving bacteremia.
I'm going to take a little bit of a detour to introduce some of the modeling concepts, and then we'll return to MRSA and how some of these tools can be used to study MRSA. But really, if there's one concept that I'd like to talk about here today, it's this idea of cell responses to their environment being multimodal. I'm going to use multimodal to distinguish from multidimensional in a way that these terms don't have precise definitions, and so this can become a bit of a mess. So just to give kind of a explanation of what I mean by this, a multidimensional response, we might call something multidimensional because we make a large collection of measurements. So for example, if we measured a large number of different cytokine responses, we might call that a multidimensional data set. What I mean by multimodality is that there's actually different categories of these measurements all made in combination. So when studying cell responses, it's quite common, especially nowadays, where we can make these very in-depth, high throughput, high characterization measurements to, say, profile many cytokines across many cell types, across time, across extracellular environments. And this just leads to an explosion of many different measurements and kind of an overwhelming amount of data. And I would say we don't have great ways of exploring, especially data in this form. And so we've been really interested in improving the ways that we can explore, model these kinds of data. So one of the tools I'm going to use in the modeling that I'm going to show you is dimensionality reduction. Dimensionality reduction I sometimes explain as being kind of magic, in that it can make everything better. If the patterns in your data follow some pattern that you can recognize using these techniques, then you can improve the ability to model the data, visualize it, explore it. Everything becomes a lot easier. And the form of dimensionality reduction that I'm going to use is a generalization of matrix factorization. So if we have our measurements in some two-dimensional form, in this case, we might have a collection of samples that have had a collection of measurements made on them, and we can factor this into sets of overarching patterns that try to approximate the original data. So we might have a collection of patterns that represent how the features are similar or different from one another. And we have a collection of patterns that represent how the samples or subjects are similar or different from one another. But in struggling with this multimodality issue, our lab has found that doing dimensionality reduction in this two-dimensional form can make it challenging to interpret the patterns that you find. And so we were quite excited when we came to realize that there are actually generalizations of matrix-based dimensionality reduction to higher numbers of modes. And so a matrix is just one example of a tensor. And so a matrix would be a two-mode tensor. If you had, say, a data set where you had subjects over time and over different measurements, that would be a three-mode tensor. And this continues on ad infinitum to many, many modes if you have the pleasure of needing to deal with data like that. Just in case I mention these terms, everything gets a little bit more complicated in tensor form, and I'm happy to talk about some of the mathematics if that comes up, because there's a lot of interesting issues with these techniques. But if I talk about, say, taking out like a line of numbers from a three-dimensional data set, this can be called a chord or a fiber. And then a slice would be like, if you slice off a piece of the end of this cube here, it's a two-dimensional grid of numbers that's a subset of the data. There's been a lot of development in generalizing dimensionality reduction techniques to multimodal forms. One example of this that I'm going to show is called canonical polyadic decomposition. This tries to approximate a three-dimensional tensor as rank 1 vectors. And so the outer product is taken from these rank 1 vectors to try to approximate this original tensor. You can think of this as kind of a generalization of PCA, in that each of these are additively independent. And so I'll refer to these as different components. And then each of these vectors tries to explain the patterns along each of the modes in this original data set. And so if we can approximate the data in this way, we can look at what these components represent, and that gives us an insight into the patterns underlying the original data set. Now, really why these methods can be I think these methods should be really widely and extensively used in particular in a lot of these cohort studies where we make really in-depth measurements in human subjects. And there's a few really great benefits that we found across several different studies now. One that I won't talk about today is this one looking at serology data, where you can actually separate out antigen, FC, and other effects. But one is that you can, in many cases, reduce the data down to be a lot smaller. So you can actually just make the data set smaller, which makes it easier to explore the patterns. You can find how the patterns relate to particular modes. So you can say if something is related to a temporal pattern, or if it's related to subject variation or something else. And then this last property lets you integrate different types of data sets. And so if you have, say, data sets of heterogeneous composition in the same subjects, you can enforce that there's some sort of common subject pattern across different data sets. And this allows you to find how different patterns are related within different types of data. And so it becomes a really powerful data exploration tool if you're trying to especially integrate different types of measurements. Now, these aren't automatically better. You have to have data that has some sort of multilinear patterns in it. And I think one of the struggles sometimes is being able to demonstrate that these are effective methods for the data sets that you're looking at. So I want to walk through kind of the simplest example that we have in the lab using biological data, and then go back to the MRSA data set and show how we can find patterns in that. And what I like about this example is that in the end, it's just incredibly simple. And so it really shows you how you can interpret the results that you get from these methods. So this is work that we've done with a biotechnology company and then have extended with ourselves and also working with some immunologists at UCLA. And it's asking the question how we can make a cytokine that selectively interacts with regulatory T cells. And this is being widely explored in a lot of autoimmune diseases. And the idea comes off of this observation that IL-2 has both a high affinity receptor form that involves this alpha receptor and a lower affinity receptor form that doesn't involve the alpha receptor. There's a whole lot of really interesting immunology here, but I'm going to simplify it down into thinking about it this way so that we can look at how we can use these tools to kind of explore and design based on data sets. Now, billions of dollars at this point have gone into designing versions of IL-2 that either bind more preferentially or less preferentially to the alpha receptor. And those have led to molecules that can be somewhat specific for Tregs. But we were wondering if you might be able to learn from the biology itself to design something that's even more selective than what's out there. And in the protein engineering that has occurred thus far, it really comes down to playing with a variety of different features of these molecules. One is the orientation of the fusion of these molecules to an antibody, which extends the half-life of the molecule. Another is changing whether there's one or two cytokines conjugated to the antibody, and then changing the affinities to the receptor. And so to really look into this in depth, because cell responses are multimodal, we created a variety of these molecules, and then we took 13 of these and we profiled them across 12 concentrations, 5 time points, 8 cell types. This quickly becomes a big and complicated data set. So it looks something like this. We're reading out how much the cells respond to IL-2 based on this STAT5 signal. This is a protein that's phosphorylated in response to IL-2. And then we want to find the patterns in it. And we want to use those patterns to design an even better molecule. Now, we, like I think many of you may think, naturally first turn to principal components analysis. It's a great way to explore data. And if you use PCA, your results look something like this. So the issue is that we had to flatten the data set. So we have to take these different ligands and doses and combine those, take the cell types and time and combine those. And so you end up with, although it's reduced the data set extensively, it doesn't really make it easy to find what the patterns are in the data set. And the issue really comes down to the fact that we've taken a 3-dimensional or 4-dimensional data set and we've smashed it down into something that's completely flat. And as an engineer, if I think about like things we might study out in the real world, I don't generally take things that I'm trying to study and then squish them down before I do any sort of characterization. So why do we do this with data sets? And so a bit of exploration led us to using canonical polyadic decomposition. This is a 4-dimensional data set. I don't really know how to visualize 4D data sets, but we draw it this way. And we use canonical polyadic CP, and then we're able to see that there's about 3 patterns in this data set. And then what's beautiful from this is that these patterns really clearly reveal the expected pattern, the patterns we'd expect to see. So for example, I'm just going to focus on this component 2. This is a pattern that we can read out from the concentration profile as being more responsive at lower concentrations. If we look at the cell type weights, it's most represented in the Tregs, and it's most represented in the alpha receptor high Tregs. It doesn't have a difference over time. And so this shows that by doing the dimensionality reduction in this way, we can really effectively figure out how the patterns relate to the underlying modes of the data set. And so to get into, just to show that this actually does make it easier to design molecules, what we ended up observing is that if we look at the component 2, the pattern we want to maximize versus the 2 patterns we don't want to maximize, that all of the more effective molecules were bivalent, meaning they had 2 cytokines attached to the antibody. And so we really simply asked, well, if 2 is better than 1, can you make 4 be better than 2? And this is work that Scott Taylor, a lab manager, has done in the lab, showing that actually, in fact, a tetravalent molecule is even more selective than what you can get with 1 or 2 attached. But that's all, again, to show that by organizing the data based on the different ways in which it's been characterized, if we can preserve some structure in the data set, then we can perform dimensionality reduction more effectively. And that also improves the interpretation of the resulting factors. So we can learn more about what we find in the end. Okay, so let's come back to MRSA. So this is the way that we kind of think about this MRSA study, in that a set of patients were collected. We're looking exclusively at the serum and plasma cytokines that were collected, as well as whole blood gene expression. And then what we want to do is compress this data down, or find the patterns in the data set that relate to persister versus resolver status. And we're going to do this by performing dimensionality reduction, then using some sort of predictive model, and seeing the association with the different statuses. It's been really fantastic to do work like this, and it really takes a big team to do a lot of this work. So I just wanted to acknowledge that there's a lot of people, even beyond Michael, who've helped here at UCLA and Duke to really enable work like this. And we're applying tensor methods in other studies where similarly, it takes a big team to collect these kind of well-characterized large scale, high throughput data sets and have all the appropriate controls and other features that provide really high quality data. Okay, so we organize this. We recognize that this could be organized in a tensor form, basically recognizing that the gene expression shares this patient axis. So it comes from the same patients. And there's one gene expression data set that can be summarized by its gene patterns as well as patient patterns. And then we have, in many cases, cytokine measurements from either or both plasma and serum. And we wanted to distinguish these along a third axis to make sure that we're recognizing that these are not prepared in the same way. And so they likely have very similar cytokine profiles, but in some cases will have differing cytokine profiles. And by organizing this as a third mode, this effectively accounts for this observation. And then our hope is that if we do the dimensionality reduction appropriately here, one, we might improve our ability to predict persister versus resolver status. And then separately, we might be able to better understand what the patterns we get in the end are. So we are looking to see whether we can improve prediction, but I think it's important to recognize that just prediction on its own isn't the end goal here. It's also to understand the resulting immunology, and that requires understanding how the prediction relates to the underlying measurements. So just like with PCA, we can increase the number of components, and we can observe that it increases the variance explained in the dimensionality reduction. So a relatively small number of components explains the majority of the variance in the data. One unique kind of surprise that happens when you use coupled forms of decomposition is that there's kind of an arbitrary scaling between the two different data sets. And so in one extreme, we can prioritize explaining the RNA data. And in another extreme, we can prioritize explaining the cytokine data. And this is something that we can choose. And so, for example, if we scale the RNA to be of greater magnitude, then this will explain the RNA data better, but that comes at the cost of explaining the cytokine data. So we have these two parameters that we need to tune, and we ended up choosing to tune them based on how well we could associate the patterns we find with persistent status. So if we vary the number of components, we see that around 8 components maximizes the variant or the association with persister status. If we change the scaling, we see that there is, in general, just increasing the cytokine scaling makes you do better in your predictions. Maybe there's a maximum, but it's really hard to say that there actually is. And then we wanted to see, well, can we do similarly or better in terms of our prediction or association with persister status? And so we performed cross-validation using a logistic regression model. And different patients had different types of data available. We had several patients that are, say, missing their plasma cytokine measurements. And that's why you have these different subsets where we've separately calculated the accuracy in assigning the persistent status. But in every case, we either see that we're doing identically to how well just the raw data does when building a logistic regression model, or we do a little bit better in our assignment with persister status. Now, around the time I was brought in, the MRSA group was just finishing up collecting a completely independent data set. And so we left that aside and we didn't touch it until we were done with our model building exercise. And we wanted to see, well, can we actually predict an entirely new cohort that we haven't built the model on? And so you can see here that actually, many of the raw data measurements did a little bit worse on when we went to this new data set. CMTF also takes a little bit of a hit. which is understandable given this is a validation cohort. But again, CMTF does identically or a little bit better in its prediction of persister status. Okay, so it seems like we have a model that is effectively predicting persistence, and then we want to break it open and see how it works. So I first just want to show these are the subject factors. And so these are the components that explain the subject to subject variation. And this is what you then build the prediction model off of. So in effect, our logistic regression model has 8 variables, and it's each of the components that explain how the subjects are similar or different. And what we saw, and we spent quite a bit of time trying to work around, but I think in the end is a benefit of factorization models like this, is that there is cohort to cohort just differences like batch effects that are being explained by the reduction. So you can see, for example, component 2 is a lot more darkly colored as compared to the separate cohort here. And so some of the components explain cohort to cohort differences. This could be differences from technical effects or from subject effects. It's really hard for us to say. But what we've found in the logistic regression model is that components 6 and 7 are very, very highly associated with resolver or persister status respectively. Component 3 is weakly associated with persister status. And if you look at 3, 6, and 7, there is a little bit of cohort to cohort differences here, but they tend to be the ones that are have less cohort specific differences. And so we can then look at, say, components 6 and 7 and start to understand what this tells us about MRSA persistence. So I just want to quickly show all of the factors just so that you get a lay of the land for this. Here are the subject factors. Here are the cytokine factors. And so we can look at these and say, for example, that GROW here is highly positively associated with component 6. Because plasma and serum were organized in this 3D way, we have a factor matrix that tells us whether a component is shared across both data types, both serum and plasma, or is unique to one or the other. And we see that both of these components are present strongly in both serum and plasma. And then finally, we have the gene expression models or modules. There's quite a few of these. So for each of these, you really have to dig in and look at what they contain. But we can do that for each of the gene expression models to understand what the gene expression data is showing us. We made one final observation that I think helped simplify how we could think about what's happening in persistence. So Jackson Chin, the PhD student who was working on this, noticed that we have 2 components that are very highly associated with persistence or resolver status, and then one that's a little bit weaker in its association. And we were wondering if we had seen this in a previous study where the dimensionality reduction was so effective that we are finally to a place where we can actually think about building nonlinear models, and we have enough data to use that as a prediction model. And so what we did is we took the logistic regression model and we compared it to an SVM model with just 2 components instead of all 3. And what we found is that if you're using a slightly more complicated prediction model, that you can basically just throw out component 3, and it's just 6 and 7 that are highly associated and are predictive of persistent status. And it's because you get kind of a little bit of curvature between the 2 components. But what's nice about 2 components is you can plot it. And so you can really nicely see here that there's one quadrant that is all of the persisters. And then the resolvers are spread out, being either high on component 6 or low on component 7. And this was a little bit surprising to us that everything would come down to components 6 and 7. Because if you look at the factor plots here, both components look pretty similar. When there's green in component 6, there's a lot of green in component 7, and vice versa. When there's a lot of purple in one, there's a lot of purple in the other. So in general, they're pretty correlated with each other. And so what we did is we said, okay, well, 6 and 7 are really the most important patterns we found here. Let's dig in and see how they're different from one another, because you do need both of them to make an accurate prediction. And because these are opposing in their effects, it's really the things that are off of the x equals y line that are going to be contributing to the prediction. And so if you just plot 6 versus 7 and look for what's off axis here, you have IL-10, which we know also already is very highly associated with persistence, IL-17 and GROW, and really just those. And then we have a couple of gene expression modules that fall a little bit further away from the x equals y axis. And digging into the cytokines as well as the gene expression modules, just to give a summary, what it really comes down to is we have a component which we think represents granulocyte differentiation, and then another component that represents granulocyte proliferation. And granulocytes are incredibly important for clearing infection. And so it sort of fits that if you're going to be a resolver, you need to proliferate to make a large number of granulocytes, and you need to have at least some fraction of those be productively differentiated and able to function. And so in the end, the patterns we find sort of fit with the biology. And something I've been struck with a lot of these tensor methods every time we've used them is that in virtually every case, we ended up coming up with very simple lessons about what the resulting signatures are. So for example, the example I used in the beginning with bivalent cytokines, it really comes down to just valency that we could improve. Here we have a very simple set of one cell type and two aspects of that cell type that together is associated with persistence. So I really think that we should be using tensor methods everywhere, and we're trying to make that happen because these come up in bioengineering all the time. It comes up in systems immunity, in cancer systems biology. Here's just a few examples of those cases where we've looked at serology data, at IL-2 cytokines, cell signaling, Jackson's paper looking at multiomics. And so I think it's important that we make these tools widely available. And these are tools that have been developed pretty extensively in the mathematics community, but haven't quite made it into being in wide use in biology. And I'm one of the lead developers on this Python package, TensorLi, that tries to make these tools easy to use, have great documentation, lead people through their analysis and interpretation. There's also reasons we haven't been able to use these tools. One example is in cohort studies, we often don't have aligned measurements. So if we want to deal with time as an axis, we would need to have people come in, say, at exactly the same day. And that isn't practical. People come into the doctor's office at different times. And so often our samples are unaligned along a time dimension. And so we've been trying to make continuous tensor methods that try to deal with this effect. Coupling is a really powerful concept in tensor factorization. And we want to try and use this more extensively, say comparing different disease types where we have the same types of measurements and be able to do more flexible coupling. And then finally, especially in immunology, in cancer, single cell measurements are becoming really widespread and common. And tensor models really don't have any solution for that because the equivalent for a tensor factorization model would be measuring this different things in the exact same cell so that we could make one dimension be cells. But that's obviously not how single cell measurements are made. And so we've been trying to do things like combine a Gaussian mixture model and basically tensorify it, like represent the mixture model as a collection of tensors so that we can find patterns in single cell data sets. And I think that will also really widely open up some of these methods. So with that, I just wanted to acknowledge Jackson in the lab did the tensor modeling for MRSA. Brian did the IL-2 work. Cyrillis is kind of our tensor expert in all things. And it's been a really great collaboration with Michael and folks here and at Duke.
So I think the big pictures are, that we're learning more about the complex and dynamic interactions between staff and the host. We talked briefly about innate memory and contextual immunity, which are not often really at the top of everyone's minds. We know there are genetic and epigenetic factors involved. Aaron did a great job talking about tensor modeling. All of this work and effort really comes down to applications that we hope might be enabled. One, of course, is that by understanding these interactions, we might identify targets of protective immunity that could be then leveraged by novel vaccines and anti-infectives. Predicting outcomes of MRSA infection would also be ideal if we could foresee, through predictive capabilities that a patient would not do well on regimen X from the outset, but then could be switched to a different regimen early. That would be great. And of course, all of this knowledge from Staph aureus may be applicable to other high priority pathogens. And I think on that note, there are a number of upcoming publications that will address some of these questions that we'll be happy to keep you all updated on. I won't go into the details now. But finally, just a note of thanks again to the NIH, in particular, Liliana Brown, Nancy Ernst, and Reed. Thank you so much for all your effort and guidance on this program. Vance Fowler, Batu at Duke, Richard Ahn, who's in industry, and then Elaine Reed, David Jertson, and Alex Hoffman at UCLA. So with that, thanks again for your time and invitation. And now, James, back to you for any questions.
Floor is open for questions. Maybe I'll start out. And a lot of the talk was on the signal analysis techniques. But I wanted to come back to some of the basic biology, which you started with, which was very intriguing to me and unfamiliar, which was your comment that upregulation of interferon gamma and downregulation of IL-17 had particular effects, and also that downregulation of IL-10 could have particular effects. And I wondered if you could tell us a little bit more about that. There are lots of people in our community who are very interested in the detailed kinetic modeling of interleukin interactions. And so trying to understand how these things are connected would be really very interesting.
Yeah, absolutely. I could share a few perspectives. Maybe Aaron would want to add to this, but there is pretty well established knowledge that IL-17 mediated pathways are really important for host defense against staph and candida. IL-17 A and F in particular activate neutrophil responses in a way that leads to chemotaxis and activation of microbicidal responses. What's so interesting is that while IL-10 is known generally to be anti-inflammatory, IL-10 in some contexts is pro-inflammatory. And that alone is an interesting point. But going back to the STAT1 story and then combining it with what Aaron talked about, STAT1 and STAT3 can create homodimers that can modulate the expression of both IL-17 and IL-10. And so one possibility that we're exploring is that the counter regulation of IL-10 and IL-17 might actually be represented in this unforeseen result, where in persistent MRSA bacteremia, we are seeing higher IL-17 levels. And that's sort of counterintuitive at face value. What we are thinking, and Aaron, please weigh in here, is that it's more about the quality of the neutrophil than the quantity of the neutrophil. And that, you can have neutrophils that are unprepared or ill-trained being released from the bone marrow in response to persistent MRSA, but they can actually do more harm than good. Let me stop there, Aaron, and get your thoughts as well.
Yeah, I think there's kind of an overwhelming number of combinations in a lot of cases. And I think that's what makes the biology hard and makes modeling it kind of hard sometimes too. Like, I know there's a lot of, there's been a lot of really great work in modeling cytokine and other signaling factor responses. It suddenly becomes really difficult to think about linking that to functions. And then even on top of that, for a productive response to infection, you often need 3 or 4 different functions. You might need proliferation, differentiation, like an activation signal, and then to overcome different inhibitory signals. And all of those relate to the signaling effects in different ways. So I think it's one of the remaining challenges in how to model all of this.
Other questions? Yeah, I can come up with another one.
I've got a really quick one.
Read, go ahead.
And then I got to jump to another call, but I always get nervous asking questions because I don't know the details of these things. But you gave some really nice overviews, Aaron, about sort of tensor modeling here and applications as they pertain to disease forecasting, resolve inverse. And I'm sorry if you mentioned this already. Can you comment a little bit on predictions for vaccine effectiveness in these? Can you, I assume it's the same application, but what's the space there in terms of predicting vaccine efficacy, since it's so hard in these AMR pathogens, escape pathogens?
Yeah, I think that the modeling techniques that I've talked about work in pretty much any cases that you have in-depth, sort of systematically characterized patient cohorts. And so we haven't really looked at any studies in vaccination that are done at UCLA. One place where we have applied this type of modeling that comes to mind based on your question is looking at system serology data from Galit Alter's lab. And that's actually what the molecular systems biology paper that I mentioned covers, is that kind of serology data lends itself to be analyzed in tensor form really effectively. And it allows you to do things like, say, the correlates of protection are related to, say, a particular antigen response, and separate that from, say, a protection being derived from a different FC property. So you could imagine doing something very similarly with other vaccine studies where you're maybe comparing, a couple different perturbations, and then do a lot of different immunophenotypic profiling and gene expression profiling and serology profiling. This is a way of finding what are all the patterns in that data and how do they relate to outcome.
Reed, I like to call this, there's a big difference between immune response and protective immune response. And that's where the challenge lies, because it's, vaccines will induce all kinds of responses, but they may not be protective.
Right. Yep. And yeah, trying to model that protection is definitely going to be a challenge of the day.
Yeah.
So you started out by making a very nice comment, which we've heard from other people in the context of viral infection, which is that where the infection is means that the immune response is different. And I was wondering if you had enough patient population that you could stratify the locus of the lesions. Did you have information about where the lesion was in the patient? Because you're collecting serum, so you're getting some kind of some kind of body average of these signals. But as you pointed out, the immune response could be very different if you had a lesion in the liver or the kidney somewhere else.
Great question. In this approach, thanks, Reed. In this approach, we excluded, as I mentioned, individuals who had complicating features such as indwelling devices or potential, loci of infection. We did that on purpose for this first stage of analyses to try to better understand, the bacteremia per se. However, you're exactly right. We know that from animal models, staph will go and hang out in the kidney even when other places have cleared. And so there's this contextual immunity that we like to think about, where there are different molecular and cellular signatures of immune response to the same organism in different places in the body. Liana Chen, who's on the Zoom now, has done a lot of work here looking at MRSA infection of skin versus kidney versus spleen versus blood. And essentially, it is as if there is a completely different organism in each of these places. And I say it that way because it is possible that there is a different organism, that the organism is adapting so much to that local context of the host that the immune system sees it as a completely different organism in many respects.
So that was one thing you didn't talk about so much. You talked about a lot. I mean, it was, but you mentioned at the beginning that the pathogen itself was something that was going to be looked at, and we know bacteria could change a lot in the way they behave depending on their environment.
Yes, this
and
work.
So
yeah,
no. And so I wondered if you did profiling, sort of epigenetic profiling of the bacteria as part of this. Well,
again, great question. The work that we talked about today focused on the host. We did look pretty extensively at the bugs, and we did whole genome sequencing. There were a couple of interesting features that came from that, and we also did phenotyping, and there were some interesting features that came of that, and I'll just summarize. Overall, if you look at the genome sequences of the organisms, and we looked at several hundred, there were no obvious gene presence or absences that associated with persistence versus resolution. However, there was a window in the genome of many organisms that had a much higher variation than you would expect. We also know from work by Ben Howden and some of the work that we've done that certain phage and their transduction and excision can cause these kinds of real-time, quickly adapting gene shuffling that might be associated with persistent phenotypes. For example, staph in the bloodstream, those that persist tend to adapt to what's called a small colony variant, which is a transient, it is not a genetically stable type of organism, but it's sort of this quick gene reshuffling so that the organism becomes low energy. It reduces many of its antigens that would have been exposed to the immune system, et cetera. And it also enters into cells in a way that is less harmful to them. So it becomes stealth by hiding, essentially. And we think that some of that gene shuffling, while it's not clear yet that there's a pattern, the shuffling itself might be the pattern. And so we're looking into how the organism might leverage that temporary genetic rearrangement in ways that promote persistence. Yeah, great question. we are, in fact, Liana is looking at epigenetics of the organism, which is something that in bacteria doesn't often get looked at, but it's an exciting, potential area of discovery.
So just, and again, please be patient with me as somebody who's not in the field. So clearance of this here is primarily due to neutrophil, macrophage, and antibody. It's not due to, we're not talking about clearance of the elimination of infected host cells, the way we would be with, I don't know, salmonella or something else. You showed some entry into tissue, but again, we're not talking about a situation in which we're talking primarily about NK cells or T cell or CD8 plus T cell clearance. Is that right?
We actually think about persistent staph in some ways as more like a virus than a bacterium, exactly for the reasons you're suggesting. On one hand, those cells, those bacteria that get into host cells and persist there in a stealth mode are very similar to viruses that we need to target the host cell, not the organism. The other interesting fact about staph is that macrophages tend to come into areas where neutrophils have responded and engulf the neutrophil and thereby kill both the neutrophil and the staph.
Yeah.
Interestingly, staph has a way of exploiting that by altering the surface of the neutrophil so that the MAC no longer recognizes it as needing to be engulfed and can temporarily live within the neutrophil. So there are these layers of evasion and subversion that staph uses that are very interesting, but have created challenges to vaccines, for example.
I mean, that I'm just cross-linking to other talks in other areas. I mean, one of the talks we heard was a long time ago was on Leishmania, where the parasite itself doesn't spread. It's picked up and spread by immune cells.
Yeah.
And so the infection is actually disseminated and caused by the immune cells rather than by the primary, the parasite itself.
Yes.
The other talk and you mentioned you're working in animal models. The other talk that struck me is potentially relevant, it depends on which animal models you're using, was some talks from England looking at modes of cell death in carnivora. And so it winds up that the response to toll-like receptors in cats and dogs is completely different from that in humans and mice. And so the mode of clearance of infected cells, infected host cells, which is what I was asking about, how much of that was important in your case, is different. And so it would be fascinating to pick an animal model that used these different wirings of TLRs and NLRs to do the kind of comparative study that you're doing here.
Agreed. And in fact, taking that idea one step further, we have used even male and female animals of the same species and the same genetic background. And staph, these in particular persistent strains actually differ in their ability to exploit certain niches in males versus females. And we see that in human disease as well. Male and female humans have more or less vulnerability to staph infection, but one in skin, the other in blood. So it's a very interesting set of variations, as you say, and it's probably a lot more complicated than we even think, and we think it's pretty complicated.
Well, any other questions? I think, unfortunately, at 4 o'clock, people can stick around, but a lot of people now have to leave at 4. But I think everybody heard the talk, so that was good. Any final question before?
No, I've just been talking with Aaron on the side. It's very interesting. I just asked him if he offered like a course in the summer, because I would be interested in doing some of those analysis on some of the works that I do.
Yeah,
But
I'll
thank
send
you
a couple
very
things
much.
along, but one thing that might be more generally of interest, yeah, the TensorLi group is thinking of putting together some more substantive tools that involve examples and other things that make it easier to apply these types of models than, you know, if you have a function documentation that's useful for running it if you already know what it is, but
Mm-hmm.
you understand the background. So yeah, there's a lack of practical knowledge on just how to recognize that these tools can be useful for your problems. So there's a few people trying to make that happen, but it takes some time.